to the next stage, which is week number 3-4. What will happen? If you say week number 3-4, then what happens is this epiblast, which is at the floor of amniotic cavity. The epiblast, which is at the floor of amniotic cavity, will form the primitive streak. And the primitive streak, you can see here, getting formed in a dorsal view of the conceptor. So you're looking from the dorsal view, means the floor of the amniotic cavity. So this is the floor of amniotic cavity, dorsal view. Some primitive streak is formed by the proliferation of epiblast. And if you take a section here, you can find the epiblast cells are migrating. Where are they migrating? To ventral. If they're migrating ventral, what is the purpose? Purpose is they will replace the hypoblast cell and take their place to become endoderm. And epiblast cells, as they replace the hypoblast, become endoderm, some of the epiblast cell will form mesoderm and the remaining epiblast cell will form ectoderm. This is called as gastrulation. Gastrulation is happening in the 3rd week of development. Let us look at some more details before you start drawing some diagrams. You notice that during development, as you find, taking a section here, notice there is an ectoderm formed by the epiblast, a mesoderm formed by the same epiblast, and endoderm formed by the same epiblast. After removing hypoblast, they have formed endoderm. So 3 germ layers are formed in the 3rd week of development by the process of gastrulation. Later you will find this ectoderm is going to form neural tube, the mesoderm is going to form cardiovascular tube, and the endoderm is going to form the gut tube. Let's have an idea of that as well. As you see, go further, there is a cephalocaudal folding, means the head comes towards the tail. If you're telling there's a cephalocaudal folding, head is coming towards the tail, take a section. As you take the section, you will notice, as I suggested, the most dorsal is neural tube, forming nervous system, coming from ectoderm. Whereas the cardiovascular tube is from mesoderm. And what about this gut tube here? The gut tube is endoderm of the yolk sac. And this gut tube is going to have one vitalointestinal duct, medial diverticulum, and one allantois, which is hindgut diverticulum. So this is all going to become a content of the umbilical cord, which we have to see in more detail. As you're moving towards all of those details, let us first talk about gastrulation in detail. Gastrulation is the formation of 3 germ layers in the 3rd week of development. And you see here, we are looking at the floor of the amniotic cavity. The epiblast cells are proliferating to form primitive streak. And remember, primitive streak will appear towards the tail end of the baby, caudal end of the baby. It starts appearing there. Towards the head end, it will migrate. So primitive streak, which is proliferation of epiblast, is happening towards the tail of the baby and moving towards the head region, which is the cephalic end. A gene is required here. It is called homeobox gene, H-O-X, homeobox. Homeobox gene is going to decide cephalocaudal end of the baby. And primitive streak is developing at the caudal end, migrating towards the cephalic end. This is all happening at the floor of the amniotic cavity. This is the amniotic cavity, and this will be the yolk sac cavity here. Remember, primitive streak will appear at day number 14. By the end of the week number 2, it will be appearing here. In the week number 3, the actual gastrulation will happen, and as you are looking at the floor of the amniotic cavity, the epiblast cells forming primitive streak and migrating into this groove towards the ventral side. As they are migrating towards the ventral side, you'll notice the epiblast cells are migrating ventral and displacing the hypoblast cell from the roof of the yolk sac cavity, taking place there and becoming endoderm. So the epiblast cells will form endoderm, the first germ layer to develop after replacing the hypoblast. These epiblast cells keep migrating ventral, then form the mesoderm. And the remaining epiblast cells which are there will become ectoderm later. The last germ layer to form is ectoderm. So first germ layer to form is endoderm. Next is the mesoderm. And last is the ectoderm. And this is gastrulation. Now once you have that idea, let's draw the diagram ourselves now. You are going to talk about the blastocyst in week number 1. So this is blastocyst week number 1. And this is blastocyst week number 2. And then you'll have the embryo week number 3. Week number 3, you should start calling the conceptus as embryo. If you are telling week number 3 is embryo, what is before that? Before that is pre-embryonic stage, which is a blastocyst stage, pre-embryonic stage. Why do you say blastocyst as blastocyst? Because it is having some blast cell and cyst-like cavity. If you say there are some blast cells, cyst-like cavity, what is the name of these blast cells? These blast cells are actually called as the inner cell mass, the embryoblast. If you say these are the embryoblast or the inner cell mass, then what is there in the outer cell mass? If you say outer cell mass, then these cells. And what are these cells in the outer cell mass? You see these cells which are there in the outer cell mass are called trophoblast. Tropho means nutrition. They are going to participate in nutrition of the baby. So trophoblast is outer cell mass and is now dividing into 2 types. What are the 2 types they are dividing into, and when they are dividing? It is when the 6th to 8th day, first week becoming second week. So first becoming second week, yes. Then number 6 to 8, yes. Then the trophoblast will divide, yes. And what do they form? What they form is, now you see, it is now dividing and ultimately will lose the cell boundaries to form a syncytium. So these trophoblast cells are dividing and ultimately lose the cell boundaries and become a syncytium, which is a multinuclear stage. If it is multinuclear stage, what is that called? That is called as the syncytiotrophoblast. Now, if these are syncytiotrophoblasts, what about the cellular cells which are intact cell membrane? If you say they have intact cell membrane, then that must be cytotrophoblast. So these are the cytotrophoblast. Yes, actually what we have seen is day number 6 to 8. Yes, the trophoblast has divided into 2, namely cytotrophoblast and syncytotrophoblast. And day number 6 to 8, there is other type of cells also dividing into 2. The embryoblast, which will form the embryo, dividing into 2. And what are the names of those cells? You will tell that the embryoblast is giving us 2 types of cell. One is called epiblast, which is dorsal, and the hypoblast, which is ventral. Now, if you say epiblast is dorsal and hypoblast is ventral, let's show them in the diagram, and you can mention here. This is the dorsal cell, which is called as epiblast. If this is the epiblast cell, dorsal cell, then you can also show the ventral. Yes, the ventral will be the hypoblast cell at the roof of the yolk sac cavity. And what is it? The floor of amniotic cavity? That is the epiblast. You can make out that this is embryonic cavity, which is dorsal, having a floor of the epiblast cell. And this is the yolk sac cavity, ventral, having a roof of the hypoblast cell. There are, the week 2 is a week of 2, means 2 cavities and 2 type of embryoblasts and 2 type of trophoblasts also. Then you will notice that by the end of the week number 2, which is day number 14 to 15, you mean to say when the second week is becoming third week? Yes, when the second week is becoming third week, day number 14 to 15, by the end of week number 2, day number 14, the primitive streak will be formed. Now if you say, by the end of week number 2, day number 14, primitive streak will form. What is primitive streak? It is the proliferation of the epiblast cell. As epiblast cells are proliferating, what will happen then? It is actually happening in the third week. Yeah, what will happen in the third week? The epiblast cells will migrate to ventral. Why would they migrate to ventral? The epiblast cells will migrate to ventral so that they can displace a hypoblast cell and take their place to become endoderm. But that will happen in week number 3, not here. And what else? What else is we have isolated some cells from epiblast by the end of second week, and those cells are called as the primordial germ cell, if you remember. So primordial germ cells, yes, if you remember, primordial germ cells are derived from epiblast, and they can be isolated from the epiblast by the end of week number 2, that you remember. And now what about the third week? Let's go to the third week now. As you are going there, you will notice the ventral layer has formed, which is endoderm, the first germ layer to develop. So the first germ layer to develop is endoderm, formed by the epiblast. Then what is happening to hypoblast? Hypoblast are actually, see here, let me magnify. Hypoblast, they were actually removed by the epiblast, and they'll be lining, I can show here, the yolk sac cavity. So you are telling they are being displaced now. Yes, actually. As the epiblast cell come here, they displace the hypoblast, and hypoblast cells will line the yolk sac cavity, forming extra embryonic endoderm. Hypoblast cells are going to form extra embryonic endoderm lining the yolk sac cavity. And who is forming embryonic endoderm? Embryonic endoderm is formed by epiblast cells. And what is the next thing epiblast cell forming? See, the first germ layer to form is endoderm, and which is the next one? That is the mesoderm. And which is the last one? That will be the ectoderm. So last layer to form is the ectoderm. Remember that the first germ layer to develop is endoderm in the embryo. Next is the mesoderm. Last is the ectoderm. Can you tell me the derivatives of epiblast here in a timeline sequence? The very first derivative of epiblast is primordial germ cell, and which is the second one. It is the endoderm, and third the mesoderm, and four the ectoderm, and five. If you say 5, I'll tell you. The epiblast cells are going to give us some cells here, and those cells are called as the neural crest cells. Remember, the neural crest cells are derived from epiblast cells, and they are called as the fourth germ layer. If you say they are called as the fourth germ layer, then what are the 3 germ layers? Endoderm, mesoderm, and ectoderm. And who is the fourth germ layer? The neural crest cell, which also came from the epiblast only. So now you know 5 derivative of epiblast. The very first to come was primordial germ cell, and second endoderm, third mesoderm, 4 the ectoderm, and 5 the neural breast cells. What do you think about this question then? Because it is second and third week, it is asking hypoblast is going to form extraembryonic endoderm? Correct. Lining the yolk sac cavity. And is the germ cell developing from yolk sac? Actually, germ cell is primordial germ cell. It will develop from epiblast. But then, from the epiblast, it is migrating towards the yolk sac. There are two things. Old edition of Gray's Anatomy says the primordial germ cell comes from yolk sac. And the new edition of Gray's Anatomy says the primordial germ cell comes from epiblast. So what do you think? Is it correct or not? It is a correct statement, but it is history. So what is the present? That they come from epiblast. Anyway, it can be taken as a correct statement, but if you are telling first germ layer to form ectoderm, no. It is the last to form. First was endoderm. So this is wrong. This is answer. What about the notochord formed by epiblast cell? Yes. How the notochord will form the epiblast cell then? Let us find out, but this remains your answer. You will notice that there is a description of prochordal plate and notochord now. The primitive streak is having some epiblast cells which are going to form notochord, and along with that we have to discuss some prochordal plate as well. What happens is when you look at the dorsal side of the conceptus, that is the floor of amniotic cavity, you will notice on the tail of the embryo what was formed? Primitive streak migrating towards the head end of the baby. And it is the epiblast cells which are going to form primitive streak. Primitive streak will have a primitive groove and a primitive node and a primitive pit. So there is a primitive streak having 3 parts, a groove and a node and a pit. Epiblast cells which are all over here, they are now migrating ventral. And when they are migrating ventral, what they do is they will form this notochord. So some epiblast cells which are the floor of amniotic cavity, they are migrating ventral to the pit, and then they will form the notochord. So notochord is coming from epiblast. Yes. And it is present between the primitive pit and the prochordal plate here, which later becomes the region of buccopharyngeal membrane also. So there will develop a buccopharyngeal membrane. Buccopharyngeal membrane ruptures to form oral opening. And here you will see the anal membrane which will rupture to form anal opening. So oral opening, anal opening, there are some membranes in the beginning. And remember, the buccopharyngeal membrane, ectoderm, endoderm is fused. At the cloacal membrane also, ectoderm, endoderm is fused. There is no intervening mesoderm here. To understand it better, let's make a diagram. This diagram is a different orientation, and what you are telling is there's a transverse section of the embryo. If it is a transverse section of the embryo, then which cavity is dorsal? Amniotic cavity. Which is ventral? Yolk sac cavity. So dorsal is amniotic cavity, ventral is yolk sac cavity, which is the germ layer, which is the cells you found at the floor of amniotic cavity. That was the epiblast cell. If these are the epiblast cells, then what was at the roof of the yolk sac cavity? That was the hypoblast. Now, as you can understand, epiblast cells are proliferating, and when the epiblast cells were proliferating, they were forming what is called as primitive streak. And primitive streak will have a primitive node with a primitive pit. Now, if you say this is going to be a primitive pit, which is in the primitive node of the primitive streak, it's an opening, and through that opening, the epiblast cells are migrating ventral. And when the epiblast cells are migrating ventral, they will go like this. And when they go like this towards the cephalic end of the baby, they are going towards the cephalic end of the baby. I told you, primitive streak appear at which end of the baby? The tail end, caudal end of the baby. And who is deciding this is tail end, that is caudal end? That is a gene. And which gene is that? That is the Hox gene. So Hox gene will decide cephalocaudal orientation. Yes, towards the caudal or tail end of the baby, will have primitive streak with primitive node and primitive pit. And through the pit, some epiblast cells are jumping, going ventral, forming the notochord. And this is the epiblast cells which will also form. They will also form the endoderm. And how do they form the endoderm? By replacing the hypoblast cell. They replace the hypoblast and form the endoderm. And they will also form the mesoderm. So they are also going to form mesoderm. Yes. And they are also going to form the ectoderm. Last layer to form will be the ectoderm. But remember, all of these germ layers would have come from the same epiblast cell. So endoderm is more ventral, and in between is mesoderm, and still higher is, or dorsal is, ectoderm, all coming from epiblast. Now you remember. Towards the tail of the baby, and there is a membrane, which is fusion of ectoderm and the endoderm. Towards the tail end of the baby, there is a fusion of ectoderm and endoderm, and that is called cloacal membrane. At the cloacal membrane, you have no mesoderm. It's a fusion of ectoderm and endoderm. Similarly, you will find there is one membrane here. What is that? That is the fusion of the ectoderm and endoderm. So fusion of ectoderm and endoderm towards the cephalic end of the baby. What is that membrane? That is the buccopharyngeal membrane. If you say this is the buccopharyngeal membrane, this buccopharyngeal membrane will rupture to form the oral opening, and it is fusion of ectoderm and endoderm with no intervening mesoderm. There is no mesoderm here. And you will also notice that there is a procaudal plate here. And this procaudal plate is thickening of endoderm. This is the procaudal plate, which is actually thickening of the endoderm and is the terminal part of the notochord. Notochord actually lies between two things. What is that? That is the procaudal plate anteriorly or cephalic end, and primitive pit posteriorly or caudal end between them. So are you trying to say this is notochord, the mesoderm kind of structure? Yes. Notochord is the mesoderm lying between primitive pit and the procaudal plate. Notochord is formed by epiblast cells and lying between the primitive pit and the procaudal plate, and it is mesodermal in origin. Later it will disappear, leaving behind the nucleus pulposus of intervertebral disc. Now if you say notochord is going to disappear and form nucleus pulposus of the intervertebral disc, what is the purpose of having notochord? It is the axis of the baby. And what is the purpose of axis of baby? It has many purposes, but one is stimulate the overlying ectoderm to form the nervous system. Remember, this notochord is important to stimulate overlying ectoderm to form neuroectoderm. So it is going to stimulate this area now. Yes. And it will become neuroectoderm. Yes, it is actually going to become neuroectoderm. And what if I remove the notochord itself? If you remove the notochord itself, then brain will not develop. It is important for induction of the ectoderm to form nervous system. If you remove the notochord, nobody is there to stimulate the ectoderm to form neuroectoderm, to form nervous system. You don't have notochord, you don't have nervous system. Primordial germ cells, as you are talking about, let me show you a diagram. I have told you primordial germ cells are isolated from the primitive streak by the end of the week number 2. So, end of the week number 2, you can isolate primordial germ cell from the epiblast at the primitive streak. Later, they migrate towards the endodermal wall of yolk sac. As they are migrating towards the endodermal wall of yolk sac, earlier we used to think that they develop from endodermal wall of yolk sac, but now it has been realized actually they came from epiblast. So primordial germ cell come from endoderm of yolk sac is old answer. What is the new answer? They come from epiblast. And what if epiblast is not in the option? Then you say the primordial germ cell come from yolk sac, endoderm of yolk sac. And at week number 4, they are migrating from the yolk sac towards the genital ridge. If you remember, we have discussed earlier, primordial germ cell should go to genital ridge in the abdomen to form the gamete. The process is called gametogenesis. So primordial germ cells should reach by the end of week number 5 to the genital ridge. And when they reach the genital ridge by the end of week number 5, they can participate in gametogenesis. What will happen if primordial germ cells are going towards the oropharynx, oropharyngeal teratoma? And what if the primordial germ cells are going towards the tail of the embryo? Then sacrococcygeal teratoma. If you remember, we have discussed sacrococcygeal teratoma, oropharyngeal teratoma are aberrant migration of primordial germ cells coming from epiblast. What do you think is the answer then? We already have told hypoblast is going to form extraembryonic endoderm. And who is forming the embryonic endoderm? Epiblast. And germ cell come from the yolk sac. Not exactly, they come from epiblast, but they also come from yolk sac, so no problem. Problem is here. Why? Because this is the first to form. It is not the ectoderm. First to form is endoderm. This is wrong, that is the answer. Otherwise, we have seen notochord is formed by the epiblast cell. Notochord is nothing but axial mesoderm. It is the axial mesoderm, but it would have come from the epiblast cells. So we'll keep this answer. Yes, you have to keep that answer.